This is a production of Cornell University.
So my name is Chris Smart, and I am a plant pathologist. I study diseases of plants. My lab focuses on diseases of vegetables, but we have also gotten into diseases of hemp. And today I am here with my student, Patrick McMullen. And and the other thing I want to point out again is that Patrick made a really nice handout on root rot issues in hemp. And so if you scan the QR code that's on the agenda, you'll pull that up. And all of the handouts from today's field day will be up online at the Cornell Hemp website, and you'll be able to find them there. So, Patrick.
Thank you, Chris. All right, good morning, everyone. So I'm a PhD student here working in Chris's lab, and I've been working with root rot pathogens of hemp. And I've been working with some root rot pathogens, soil-borne pathogens of hemp. Most recently with Pythium. I'm sure a lot of you have dealt with Pythium in seedlings and damping off issues in greenhouse and outdoor production. So Pythium is an oomycete pathogen that causes damping off and early seedling death. The swimming oospores really like the water and damp soils, and you'll experience this disease a lot more in those cool, wet climates. So in this experiment I'm running now, we have these two flats of seedlings. These are 2-week-old seedlings. The one that Allie over here is holding are just healthy 2-week-old seedlings. Chris's over here, they're dead. There's nothing there. So these
Chris,
were
can you point it over here, Chris?
Yeah.
Can you point it over here?
Yep, so take a look as they bring these flats around and just look at the extreme difference there of what Pythium can do to seeds. My current research interest here is testing control strategies of Pythium. We all know that there's very few labeled products for hemp right now, and all of that's currently in the research process and to see what's efficacious and what is approved. So here I tested some different seed treatments. I had 2 copper products and a phosphite product, and as you can see in Chris's flat, none of that seemed to be effective. But of course we're still in the process of testing more seed treatments to help prevent these damping off issues.
They're very photogenic.
Yeah.
Why don't you explain the experiment, the inoculation, what's inoculation?
Yep. So to do this, I planted, these are just styrofoam flats with 128 cell flat, all one seed lot, and they were direct seeded into potting soil. And then these trays were placed in a tub of water and just allowed to float on top. Tobacco is often started in a similar method to this. So then what I did in the inoculated group was I had petri plates of Pythium growing, and we scraped off the spores from those plates and dumped that inoculum into the water. So the spores the swimming zoospores could enter the roots and the soil seedlings as they're germinating, and you can see that in this tray here there is practically no root growth coming out the bottom. And then in Allie's over here, there's all of those healthy white roots. So other signs of Pythium that you'll often see if you do have plants that emerge more are kind of a browning of the stem as it does damp off. And brown roots are also quite common. And this is a commonly observed disease in greenhouses and hydroponic production, really anywhere where there's a lot of water. And it can easily be transmitted if there's any kind of splashing of that contaminated material. So right now we don't really have good chemical control strategies, but of course you could employ some good cultural practices, good sanitation methods between your cropping seasons and cycles, and starting with clean planting material. So those are kind of the best information I could give you for Pythium right now. I'm also interested in Fusarium, which is another soil-borne pathogen, which causes some similar disease symptoms. You'll often see wilting and yellowing on larger plants and eventual plant collapse and death. On young seedlings, you can see damping off, similar to what we saw here with Pythium. And we're testing some of the same control strategies for those right now as well. If you scan your QR codes on those on your schedule, there's a more detailed fact sheet about Fusarium and its life cycle. But similar to Pythium, it also likes those cool, wet soils, and it's really an environmental fungus in the soil that persists for a long time. So it's important to have healthy seedlings established that can withstand these soil-borne pathogens and really encourage early brooding. And since these are all soil-borne pathogens, our lab is also interested in the soil health and the microbial communities surrounding the roots. So I'm currently involved in a soil microbiome study where we're analyzing and sending off samples of root cores that are around the growing hemp plants in the field to analyze what microbes are present throughout the growing season, both at transplant and then as we progress closer to harvest. So we're interested in what the microbial composition is, what kind of bacteria and fungi are in that area, and how this changes over the season, and how we can use this information to better inform our practices about growing strategies and controls of diseases and pests. There we go. And Chris, do you have more to add?
I always have more to add. I will say that if you see damping off in your greenhouse or in your field, we would actually be happy to get samples. We want to know which Pythium species. So Pythium, as Patrick said, is a water mold. It loves water. But here in New York or in the Northeast, what are we dealing with the most? Because if we're going to be looking at seed treatments or cultivars that may have some resistance, we want to be using the pathogen that you guys are dealing with most frequently. And so anything you can get to us, that will go right into our screen just to see, oh, is it more aggressive, is it less aggressive, what's most common that we see, or is it just a total mixed bag depending on what Fusarium or Pythium you may have on your farm. I'll also say that soil-borne diseases generally are worse in heavy soils. Here in Geneva, they call our soil loam. I think they're using that word remarkably generously for the soils that we have. We do see a lot of puddling, we see some root rot, and just it's not a well-drained soil. Anything you can do to increase the drainage, as Patrick said, will certainly enhance the health of the plant and reduce the possibilities of root rot. And then the other thing, I was talking to a few people beforehand, is that hemp is also just very susceptible to flood damage. So sometimes plants will look like this, and they simply have drowned. They're not very good at holding up in standing water. So again, anything you can do to enhance the drainage of your soil will certainly help. And the final thing that I always say when I'm at extension meetings is that while Patrick, as well he should be, is very pleased with this experiment, and we smile a lot about diseased plants. This is a research experiment. We are killing plants in our research labs so that we can learn more about what's happening on the farms in New York. We hope that you never see anything like this, but if you do, let us know, because we want to make sure that we're helping everyone that has root rot problems. So, and this afternoon, we'll be talking about powdery mildews, which is also another issue here in New York, and that will feature Allie. So, questions? Yes.
What are you doing genetically? Are you doing any plant breeding for pathogen resistance, both in soil and for controlling weeds, like they have Roundup Ready soybeans and such?
So the question is about breeding strategies right now. Are we breeding for resistance to diseases? For Fusarium and Pythium, not currently. We don't know if there's genetic resistance. That's further research projects that could take place, but we just don't have that information currently available. In other pathogens like powdery mildew, which if you come to the afternoon sessions, there are breeding programs taking place here for powdery mildew resistant hemp. So that's currently ongoing work, and there's optimistic results with some of that.
What about weeds?
And weeds?
Yeah, so for weed control, as you were saying, glyphosate-resistant plants, we have Faith Hatt, who is here somewhere, has been working on hemp transformation and has both tissue culture and transformation up and running. We currently do not have any plans to put ourselves herbicide resistance genes into hemp. That we're really focusing on identifying cultivar, figuring out what the issues are, tackling some of the larger issues, rather than focusing on the regulatory hurdles that come with genetically engineered crops. At least, the breeding labs can talk more about that. Actually, I don't know their plan. They are looking at genetically engineered plants for research purposes to control out flowering time and other areas. But to my knowledge, there's nothing that's focused on herbicide resistance.
Thank you.
In soilless media, do you notice the diseases more or less in a cover floor versus in the ground? It has better drainage?
Yeah, so that's okay. The question was, do we notice these diseases more readily in different soilless media versus in the ground soil out here, or differences between greenhouse media we might be using? The true answer is we don't have the data to support whether one's better than the other. But of course that these pathogens really like the damp, wet soil. If you heard Neil Madsen presenting earlier about the importance of drainage in your potting mixes, you do want that high air porosity, so you're not saturating the plants and making sure that they have good drainage. Those are kind of the best strategies. Yeah. So anything with high air porosity. Question. So this was the question was what are our potting mixes that we used here. So this is a peat potting mix that we just use in our greenhouse days here. We didn't sterilize it before. Often if you do autoclave your soil before planting, it's just more difficult to grow things. There are beneficial microbes that are just always present in the environment. But we didn't add anything else to these seed treatments. It's just our peat-based potting mixes. No. Not mycorrhizal enhanced. Yes, Neil?
Could you add temperature that's going to be like this? It's like warm temperature, so cold and wet together is like a bad combo.
Yes. So Neil is talking about environmental temperatures of the roots. So cold and wet are kind of your worst case scenarios for growing hemp. Roots like warm temperatures and well drained. Of course you need an adequate amount of water, but you don't want them saturated, and you certainly don't want the root zone cool. All right.
Thank you. Nice job, Doctor.
Should he hand that?
This has been a production of Cornell University. On the web at cornell.edu.